Good morning, everyone. Thank you for all of you attending our early morning seminar this morning on research that's happening in New York State. I'm Jo Wiederhorn. I'm the president and CEO of the Associated Medical Schools of New York. And I'm going to keep this very brief. We have a very short time frame. I do just want to give you a little bit of housekeeping. When you have questions, please put it into the chat. Dr. Taubman will be monitoring the chat room and will be asking questions at the end. So now I would like to welcome Dr. Augustine Choi. Dr. Choi is the Suzanne Dean, the Dean of the Weill Cornell Medical School. I am so sorry, Dr. Choi. I really is the Dean of the Weill Cornell Medical School in Provost, and he is going to take it from here and introduce Dr. Fauci.
Thank you, Jo. Good morning, everyone. We are so delighted and thankful to Dr. Fauci for joining us today. Dr. Fauci doesn't need an introduction, but I'll do a brief introduction for Dr. Fauci. Dr. Fauci is a New Yorker, born and raised in Brooklyn, attended the Regis High School here, Upper East Side, not so far from here, where I'm sitting, where he, after graduating from College of the Holy Cross, he attended medical school at that time, Cornell University Medical College, now Weill Cornell Medicine, where there's a rumor that he graduated first in his class, in the class of 1966. He completed his internal medicine internship and residency here at the New York Hospital Cornell Medical Center. And after his chief residency, he joined the National Institute of Health. Dr. Fauci has so many honorary awards. I will be very brief. He's a member of the National Academy of Sciences, the National Academy of Medicine, the American Academy of Arts and Sciences, and was awardee of the Presidential Medal of Freedom, the highest civilian award. He is currently the director of the National Institute of Allergy and Infectious Disease and has been an advisor to seven US presidents. And we all know he's currently the chief medical advisor to President Joe Biden. Thank you, Tony, for all what you're doing for us to keep us safe. And I'll turn over the podium to Dr. Fauci. Dr. Fauci.
Thank you very much, Augustine. It's a real great pleasure to be with you all here this morning. I'm going to give a very brief, about a 10-minute introduction to talk about, as it's shown on this slide, innovation in the time of COVID-19 and fundamentally the biomedical research response, much of which is done by the academic community, including the very robust academic community in the New York City area. So the work that has been done literally over the last year and a couple of months is most extraordinary. If one does a search in PubMed, well over 100,000 COVID-19 related papers have already been entered as of February 10th, 2021, which was yesterday. If one looks at the biomedical research response, I've broken it down and I'll go very quickly on each of these into 7 distinct areas. The first is the epidemiology of this most extraordinary historic pandemic that we're all experiencing in. Only in January of 2020 was it recognized that we were dealing with a novel coronavirus when the Chinese put on a public database a virus isolated from one of about 20 individuals who were identified with an unusual pneumonia in the Wuhan district in central China. Fast forward to where we are today. As of a couple of days ago, there have been 107 million cases and over 2.3 million deaths globally, making this the worst pandemic in the last 102 years. Unfortunately for us, the United States is the most seriously impacted country with about 27 million cases and over 460,000 deaths. There are so many aspects of this pandemic from an epidemiological standpoint that are very interesting, unfortunate, and unique, including the extraordinary disparities racially and ethnically, with brown and black people suffering disproportionately both in numbers of infections and in serious outcomes. Very quickly, the virology. We've studied coronaviruses with regard to the common cold for decades now, including the work on SARS and MERS. But a quick snapshot, this is an RNA virus which replicates and mutates very readily. There are 4 structural proteins, the most important of which is the spike protein, which binds by its receptor binding domain to the ACE2 receptor cell, which is seen in the upper and lower airway, in the GI tract, and even in cardiovascular tissue. This spike protein is the fundamental basis for virtually all of the vaccines that are currently either in use or being tested. With regard to transmission, well established that this is a respiratory-borne virus, but the interesting thing, and again, one of many unique things about this virus, is that about 1/3 to 40% of all infections remain completely asymptomatic. We've never seen anything like this with any respiratory illness. But superimposed upon that is more than 50% of all transmissions occur from someone who is either asymptomatic and will never become symptomatic, or someone who is pre-symptomatic and will be going on later to symptomatic disease. We now have a number of diagnostics. The one used most frequently for specific diagnosis is the PCR molecular test, highly sensitive and highly specific. We're currently moving towards antigen tests, which can be used much more for surveillance, less sensitive, but nonetheless very useful, particularly if used repetitively. And then finally, of course, antibody tests, which indicate a person has been exposed and infected. When you look at the clinical course, as I mentioned, anywhere from 1/3 to 40% of all individuals never develop any symptoms. But those who do, about 80% have mild to moderate symptoms that do not require any specific intervention, such as hospitalization. However, somewhere between 20 and 25% who do have symptoms have either severe or critical symptoms, leading to a case fatality that can range anywhere from a couple of percent to up to 20 to 25% for those who require mechanical ventilation and or high flow oxygen. If you look at the severe manifestations of COVID-19, this is of course dominated by the acute respiratory distress syndrome. But the more that we've learned about this perplexing disease over the months of our experiencing with it, we know now that there's a considerable amount of other organ system dysfunction, from neurological to cardiovascular to renal, with unusual pathogenic mechanisms such as hypercoagulability and hyperinflammation, often in the later course of the disease, leading to the complications and often death of individuals. Children, to some extent, can go on to develop, usually a week or two after infection, a multisystem inflammatory syndrome that is very reminiscent of Kawasaki disease. And recently, some adults have been shown to have this syndrome. The people who are at increased risk for severe illness is older individuals with a much greater hospitalization rate per 100,000 people, as well as people of any age with underlying medical conditions, which we refer to as comorbidities. Prominent among these are morbid obesity, cardiovascular disease, chronic lung disease, and diabetes, among others. There have been a number of therapeutics that have been developed over the past several months, some of which have been approved by the FDA, like remdesivir, and others, such as a variety of monoclonal antibodies and convalescent plasma, which have received emergency use authorization. Also, a number of other interventions are in the process of various clinical trials, such as hyperimmune globulin, direct antivirals, anticoagulants, etc. One of the most useful therapeutics for advanced disease, interestingly, is the commonly used anti-inflammatory agent dexamethasone, which is shown to significantly diminish 28-day mortality in people who are hospitalized on ventilators or requiring high-flow oxygen. Probably the most important goal that we have is to develop, by targeted antiviral therapy, the kinds of direct antivirals that we have used so successfully for HIV with antiretrovirals, as well as for hepatitis C. This obviously will take a fairly long period of time when you think about targeted antiviral, but we feel it's well worth the effort. Finally, with regard to vaccines, that is the success story in this extraordinary pandemic, with record-breaking time and results associated with the development of vaccines. The US government has invested heavily in the development and/or facilitation of the clinical evaluation of a number of vaccines representing 3 separate platforms: nucleic acid in the form of mRNA, adenovirus vectors, both chimp and human, and recombinant proteins. As you all well know, 2 of these, the Moderna and Pfizer-BioNTech, have now received emergency use authorization with 94 to 95% efficacy and a good safety profile. I must point out something that is really striking and will become historic, is that from the time that the virus was first sequenced and shown in a public database on January 10th, to the time they had a vaccine that actually went into the arms of individuals, was less than 1 year, which is over and above beyond unprecedented, since something like this in prior years and decades would likely have taken several years to accomplish. A testimony, again, to the work that's done in so many medical centers, including those in the New York City area, due to the extraordinary scientific advances, for example, in vaccine platform technology. I want to close with just a comment on efficacy versus effectiveness. As you know, the vaccines are efficacious to the tune of 94 to 95%, but in order for them to be effective, we need to make sure that we vaccinate the overwhelming majority of the population. And as shown on this slide, there still are concerns about everything from long-term effects, side effects, and safety and effectiveness, which we need to reach out to the community to show them, in fact, that we are dealing with a highly efficacious and very safe vaccine. I want to close with this last slide of something that we're all striving for, given the history that we've gone through with now the 3rd coronavirus pandemic, that we have to put a truly full-time effort with a lot of resources on developing a universal coronavirus vaccine as a major high priority. I'll stop there. Thank you very much.
Thank you, Dr. Fauci. On behalf of all of us, thank you for keeping us safe. And again, thank you so much for your time. Thank you.
Thank you, Augustine. Good to be with you.
Thank you, Tony. Okay, so I now introduce to you Mark Taubman, who will moderate the rest of the scientific sessions. Dr. Taubman is the CEO of University of Rochester Medical Center and the Dean of the School of Medicine and Dentistry at the University of Rochester. He is currently the chair of AMSNY. So Dr. Todd, I turn over the podium to you.
Thank you very much, Augustine. And I also give my thanks to Dr. Fauci. We're going to have 4 wonderful speakers today, and I will introduce them each right before they speak. The first is Dr. Liise-anne Pirofski. She is the chief of the Division of Infectious Diseases and the Jacques and Selma Mitrani Chair in Biomedical Research at Albert Einstein College of Medicine and Montefiore Medical Center. Her research programs are focused on vaccine and antibody-mediated immunity and the development of antibody-based therapeutics for infectious diseases. Dr. Pirofski.
Thank you very much for that introduction, the honor of addressing this session this morning. And I also want to thank Dr. Fauci for his extraordinarily inspiring leadership. It really means a lot to us in the scientific community, and it lifts us up every day. So we are 1 year into the onset of this new disease that was described very beautifully by Dr. Fauci. And just 1 year after its recognition, we are in a phase where we've recognized that this has been an absolute humanitarian catastrophe from the beginning. We have lost 460,000 Americans to this disease, which has, as was pointed out, a very unusual pathogenesis that I will just underscore a couple of aspects of which. This viral phase of disease, which is the first phase of disease, as Dr. Fauci mentioned, is largely asymptomatic, and the majority of people, those who are unlucky, progress to develop a disease that then largely becomes dominated by the inflammatory response. And that inflammatory response involves the lungs, primarily, other tissues, as was mentioned, and can wreak havoc for the coagulation system, as well as a very dysregulated immune response. And the development of therapies has focused very much on trying to control these two phases of the disease. Steroids, as were mentioned, have been effective, remarkably so, in patients who are severely ill and requiring oxygen. In the viral control phase, in the early phase of the disease, remdesivir is approved for use and has been shown to improve outcomes in terms of getting better faster in patients that are early in disease. And I'm going to focus on these antibody therapies and tell you about our work with convalescent plasma in the Bronx. I will just mention that the monoclonal antibodies have had authorization for outpatient use for a couple of months now, and convalescent plasma has been authorized since August. Why convalescent plasma? Well, this is a modality that was actually our inaugural form of antimicrobial therapy, and it has been deployed pretty successfully early in the 20th century against a number of epidemics that arose then, and has been used in each of the respiratory pandemics of the 21st century. The idea is pretty simple, that when a person recovers from disease, that their arms, their bodies, are factories for a very rich and diverse group of antibodies that recognize the different portions of the virus, and that those antibodies have the capacity, we hope, to be able to maybe prevent disease or treat disease in patients that are ill with COVID-19. Sorry. So I've worked on mice. I am a physician scientist that works on antibody immunity for the past 30 years. And when the pandemic hit, both wearing my hat as infectious disease chief, trying to organize our response at Montefiore, as well as a scientist, I felt like this was the moment to get the band together and to see whether we could leverage antibody therapies for the treatment of patients in our community. And this band included scientists at Montefiore and Einstein, clinicians, and our community, who very bravely and courageously came forth and donated their plasma so that we could develop assays to measure the antibodies in their plasma and measure its functional activity, and plan a couple of studies that I'm going to tell you about now. So our first study was undertaken in April and ended in early May. It was pioneered by this very talented young clinician investigator, Hyuna Yoon, who you see on your screen, who was able to publish her results very recently. We took plasma from our donors in our community. We tested it to see whether it had antibodies. We delivered it to patients in our hospital who had respiratory evidence of disease. These patients were actually severely ill. Please remember that in the spring, we were overwhelmed with patients at Montefiore, and most of the patients who were hospitalized were severely ill. We looked at the outcome of these patients in terms of mortality at 28 days by matching them to patients that had similar characteristics at the time of admission. And what we found was that overall, across this entire cohort, there was really no evidence that plasma was effective. I don't know why the slides are changing. Yeah, sorry. But what we found in this analysis which Dr. Fauci mentioned and which is very well known, is that age was a very important driver of prognosis, severity, and death in our patients. And so when we look back at our data and stratified it by age, we found that those who were less than 65 had really a remarkable improvement. When they received plasma, they had about a threefold reduction in their mortality. They survived better, and we basically concluded from this that age may be an important determinant or severity of plasma effect. So we're 1 year into the pandemic, we're sort of rounding third and heading home, and we know a lot about plasma. We know that it's safe. It's been given to well over 100,000 patients in our country and across the world, and it's shown signals of efficacy in case control studies and observational studies, and all of those things indicate that it's having potentially a beneficial effect. And most importantly, for our overwhelmed hospital populations, is that it has shown evidence of reducing mortality, particularly when it's given early and when it's given in high titer. But there are a lot of things that we don't know yet about plasma. We really don't know its efficacy versus a control. And this is important because the standard of care has changed drastically since the beginning of the pandemic. And we really need to know the efficacy of plasma as a function of our patients, particularly in the Bronx, where we have, we're the, epicenter of the pandemic in New York City and the US for a while, and we may be again. And we really need to understand how racial and ethnic disparities, how age, and how comorbidities and concomitant therapies, which have changed a lot, may affect the efficacy of plasma. And we also need to know more about the plasma itself. We know we need to give a lot of antibody, but we still don't really understand exactly what the antibody is doing and what it needs to be like. So in an effort to answer these questions, we began a randomized control trial of convalescent plasma in the Montefiore population. We collaborated with NYU Medical Center and had the blessings of our two centers, and on a wing and a prayer began this study in April, mid-April, and recruited about 150 patients, at which time we ran out of patients. We flattened the curve here in New York. At that time, the NIH became interested in our study, and through the generosity of their support and an amazing program manager there, through the CTSAs, we have massively expanded this trial to a number of other centers. We've now enrolled over 870 patients of a target of 1,000, and we will be, I hope soon, being able to understand whether our plasma worked. Using this WHO ordinal scale, I want to again shout out these very talented young women who are really the future of research in our field of infectious diseases. One more point about our trial, while we're waiting for the results, is that we're very proud that we have enrolled patients in the proportion that they are in the Bronx. The Bronx is a very diverse borough. Over 80% of the patients enrolled in our trial are underrepresented minorities, and we are just completely honored that our patients trust us and they have agreed to participate in clinical research. It is absolutely essential that we include patients that are the most disproportionately affected by this disease in our trials. So I will just finish by saying that research at Montefiore and all across New York City during this has really galvanized many aspects of academic medicine. It has really celebrated the importance of research, and honestly, it's the reason that we get up every day, both in terms of the hope of finding help for our patients and also honoring the participation of our community in the effort to bring an end to this pandemic. Plasma, as I mentioned, looks like it is demonstrating some efficacy, but we really need to prove this scientifically. That's what this is all about. That's why we're doing this. And hopefully it will help us informed guidelines for clinical care. This is a very scarce resource. We need to use it properly. And honestly, as Dr. Fauci mentioned about these recurrent coronavirus pandemics, we really need to be prepared for the next pandemic. With that, I just really want to thank everybody who has participated in this research, the support that we've gotten, and say thank you to everybody.
Thank you, Dr. Pirofski. Our next speaker will be Dr. Todd Evans. He is the Peter I. Pressman, MD, Professor of Surgery, Vice Chair for Research and Chief of the Division of Research and Surgery, and the Associate Dean for Research at Weill Cornell Medicine. His research programs are focused on the molecular regulation of organogenesis and the underlying genetic and epigenetic programs that are involved in developmental defects and organ-based diseases. However, in 2020, his laboratory pivoted towards COVID-19 research using human pluripotent stem cells to discover drugs that block SARS-CoV-2 infection of various tissues. Dr. Evans.
Thank you very much, Dr. Tallman. It is an honor to represent my AMSNY colleagues who have contributed so much to our current understanding of COVID-19 biology, as I hope everybody gets a flavor for this morning. And also to represent Weill Cornell Medicine, where we are, of course, very proud to be the alma mater of Dr. Anthony Fauci. I'll just mention that I have no conflicts of interest for what I'm going to discuss. Now, in mid-March last year, as the pandemic was surging in New York City, our hospitals were being swamped and much of the normal research activity was being shut down. So a number of us, including stem cell biologists like myself, who is not a virologist, started thinking about whether there was an area where we could contribute based on our research expertise. So could I have the first slide, please? Now, it seemed to us that there were a few relevant challenges, as actually both of our, Dr. Fauci and Dr. Pirofski have already mentioned. First of all, the model systems being used to study the SARS-CoV-2 virus seemed potentially inadequate to fully understand the disease. They typically were not using human cells, or if they were using human cells, they were abnormal cancer cells, unlike those cells that would be infected in our patients. Second of all, we really had no idea why patients seem to have such diverse symptoms. As has been mentioned, many patients are often even asymptomatic or have mild coughs, but of course sometimes progress to horrific outcomes involving many organ systems. Next. So we considered that at least part of the problem was that we knew little about what cells were actually being infected by the virus. Now clearly it's a respiratory disease, so the lung would be a primary target. But the lung is a very complicated organ with many different cell types. And in addition, all these other symptoms suggested potential involvement of other organ systems, including, for example, the gut and the heart. So as stem cell biologists, our normal day job is to generate all types of cells and to study a variety of human diseases, such as diabetes, heart disease, or colon cancer. And so we decided perhaps we could adapt our research using these cellular tools to study instead this infectious disease. Next slide, please. Fortunately, we were in a good position to make this pivot because we have had, over the years, received excellent support from the New York State Department of Health through their NISTEM program that supports stem cell-based disease research. So I want to take a moment to thank the New York State legislators for recognizing the value of that program and for your support. It's made a huge difference in our ability to do this research. And so we did exactly that, and we started generating many different cell types with the following in mind. Next. First of all, that normal human cells may better represent the response to infection experienced by COVID-19 patients. And we can look at many different cells. We can evaluate to see whether or not they, in fact, can be infected by the virus, and if so, how they respond. And finally, can we try to adapt such a platform to screen for drugs that block or inhibit virus activity? Now, I'm sure you've heard the saying that it takes a village to raise a child. Next slide. Well, we learned quickly that it takes at least a city to take on a pandemic. So we built a research consortium, largely under the driving force of my colleague, Dr. Shubing Chen, with a number of our colleagues at Weill Cornell, such as Robert Schwartz, and many others, including many students and postdocs, and most importantly, critical partners, such as the famous virologist David Ho up at Columbia and his team, and the internationally recognized expert in coronavirus, Dr. Benjamin Tenuver at Mount Sinai, and his team. So at the exact time, in the depths of the initial New York City surge, March, April, May, as much of the city was being shut down, a large team of New York City biomedical research scientists were traveling up and down Manhattan, working long days, 7 days a week, working together spontaneously, trading reagents and expertise to learn as much as we could about how different cells respond to viral infection. And I know that researchers across the state were doing the same thing, actually across the world. So let me give you one example of how this works. Next slide, please. Shown here are tissue sections on microscope slides from a colon biopsy of a COVID-19 patient. Now the brown staining that the arrows show in the middle panel shows us the presence of SARS-CoV-2 RNA. So we know that indeed the colon of this patient is infected by the virus. And in fact, if on the right, it's a high resolution picture with arrows, you might even be able to pick up small actual viral particles. So what we learned is that the colon is an actual direct target for viral infection. It's likely that that's at least contributing to the gastrointestinal disease of these patients. It's not just an indirect side effect. And then the question became, well, can we model this in a Petri dish? Next slide. Now, in the lab, we can use stem cells to generate colon mini organs, or organoids, that can model the human colon. Now, it's a relatively simple model, but it includes many of the cell types and the structures that are present in the normal human colon. And indeed, we found that these organoids, these colon organoids, were readily infected by the virus, and that they respond very much like what we see is happening in the colon tissue of COVID-19 patients. For example, in terms of the genes that they express once they're infected, and how that infection interacts subsequently with the immune system. Now, this is a very high magnification close-up of a single organoid, which they're actually very small. Next. But with this technology, we were able to grow hundreds of thousands of these organoids and then place them into individual wells of a multi-well petri dish, which you see sort of on the left side. This is a 384 well dish. So we put the organoids in each of those wells. We add virus to each of the wells. And then in addition, we add individual FDA-approved drugs to each of the wells. So one drug in the first well, a different drug in the second well, etc. Now, after a day or two, we can read out how much virus had infected the organoids. So those on the right, the blue dots, each blue dot represents a specific single well. And of course, by that, it represents a specific drug. Now, as you can see, most of the organoids were very much infected. There's a lot of viral activity. But in some wells, there's very little virus activity. If you look where the red line is, those wells that show activity below the red line indicate that the drug had blocked infection. Next. Now, here are two examples of drugs we found to be effective against SARS-CoV-2 infection. One is called imatinib, and it's a frequently used cancer drug. And the other on the right is called mycophenolic acid, which is used commonly to suppress the immune system in transplant patients. Now, the key to this is since our screen was for FDA-approved drugs, we can be quite confident that they would be safe to use by COVID-19 patients. And in this manner, we may be able to repurpose these drugs, not for their initial use for transplant patients or cancer patients, but for COVID-19 patients. And we've carried out similar screens with other tissue and organoids, such as cardiomyocytes of the heart, lung organoids, and we've, for example, found in the lung organoids, these drugs are also very effective at blocking SARS-CoV-2 reinfection. Finally, in the next slide, in summary, what this consortium was able to find is that, yes, human stem cells can be used to model SARS-CoV-2 infection and model in many ways COVID-19 in a simple way. Many, but certainly not all, of the tissues generated from stem cells do support infection and replication of the virus. And they respond similarly to the cells that get infected in COVID-19 patients, which gives us confidence that these are good models. And the system can be used as a platform for discovery of drugs that block infection or activity of the virus, and therefore provide good candidates for repurposing for clinical trials. And I'll stop there. I'll just thank our team, as I mentioned, particularly our core team, Dr. Shubing Chen, Robert Schwartz, David Ho, and Benjamin Tenuver, and all of the students and postdocs that have worked with us, and funding from the NIH, Bill and Melinda Gates, and certainly the New York State Department of Health. Thank you.
Thank you, Dr. Evans. Our next speaker is Dr. Stephen Thomas, a professor of medicine and professor of microbiology and immunology and infectious diseases from SUNY Upstate Medical University. He is the chief of the Division of Infectious Diseases and director of the Institute for Global Health and Translational Science. Dr. Thomas specializes in the study of viruses and vaccine development. He played a key role in the US government response to the West African Ebola outbreak and the MERS, COVID, and Zika epidemics. And he's currently the coordinating principal investigator for Pfizer's phase 2/3 COVID vaccine trial. Dr. Thomas.
Thank you very much, Dr. Talman. Good morning, everyone. I'm going to speak this morning about leveraging clinical research capabilities to support COVID-19 vaccine development. Next slide, please. So the bottom line up front is that develop strategies. So for example, therapeutics or actionable knowledge products or vaccines. So to develop strategies to defeat COVID, the entire biomedical research community, which you've already heard, we really had to coordinate a major mission pivot. And the ability to adapt and to pivot like the community has had to do, it really requires sort of foundational institutional expertise, subject matter expertise, experience, combined with robust research support infrastructure within the institution. And my institution at SUNY Upstate have been making long-term investments in trying to acquire the right people with the right subject matter expertise in clinical research, and also building clinical research capabilities. And it was through that strategic investment that when the opportunity came to participate in trying to develop a vaccine solution to the pandemic, we were able to answer that call. Next slide. Thank you. So I want to start basic. What is a physician scientist? You've heard that term a couple of times. So, first we are clinicians, and so we care for people. We help these people try to prevent the onset of disease, but we also try to help them in a more holistic way to achieve mind, body, spirit wellness. Now, in the event that someone does become ill, we try to diagnose what the cause of that illness is, and of course, provide a treatment plan which will get them well again. But the other thing that we do, which may not be known to everyone, is that we are sentinels on the wall. We are constantly observing events. Where I live in central New York, opioid use disorder or Lyme and tick-borne diseases, these are events that we have seen evolving over time, and that because we have been observant, we've been able to raise these as health concerns for the region. And this is very much what happened with the physicians and scientists in Wuhan, China. And I put the ProMed post there from the end of December of 2019, where there was an observation of a cluster of pneumonia cases that was increasing exponentially without an identifiable cause. And this was sort of the shot that kind of heard around the world that let us know that we were on the front end of a pandemic. But in addition to being clinicians, we're also scientists. And so we ask questions and we try to answer those questions. We develop hypotheses to try to explain the observations that we're making, and then we design and conduct experiments so that we can generate facts and we can generate knowledge to try and either refine those hypotheses or to say, this is fact and this is why what we are observing, why it's occurring, and how we can potentially combat that. So I look at physician scientists as working with basic scientists and research support personnel to take knowledge and advance it into some kind of tangible solution, whether it's convalescent plasma or a therapeutic or a vaccine candidate. Next slide. So what are clinical research capabilities? So clinical research in general is research involving humans. And it may seem on the surface as a very sort of straightforward operation, but I can assure you it's incredibly complex. And it's complex first and foremost because it really requires a team of teams. And this is, the people are the most valuable resource, but unfortunately they're also the most elusive to find and to gather and to get the right team together. If you want to conduct a clinical trial, let's say a vaccine trial, for example, you need clinicians, You need research coordinators. You need people who can work in the laboratory, folks who can recruit volunteers. You need people that know how to prepare a vaccine, folks who know how to ensure that you remain compliant, and people that are providing oversight to ensure that the trial is done in the most high quality manner. So team of teams. But you also obviously need physical infrastructure. So you need a place that you can actually see volunteers. And if they become sick and become patients, a place that you can diagnose and help to treat the patients. And then you need a robust infrastructure to be able to collect human biologic samples, for example, blood or a nasal swab. You need to be able to collect it, process it, keep it cold and store it and ship it, and then ultimately get it to a laboratory so that you can create the data that's going to allow you to know whether this intervention worked or did not work, or it's safe or it's not safe. And then, although maybe less exciting, incredibly important are the research support personnel. So the people who manage the contracting actions that are required, or finance, again, regulatory compliance. So it's a very complex operation. Next slide. So what was our pre-COVID mission at SUNY Upstate? So we had a very diverse portfolio of trying to develop medical countermeasures, vaccines, drugs, diagnostics, actionable knowledge products. And we worked with many different partners. So we worked with the Department of Defense, trying to protect service members who were deploying to places where dengue and malaria and Zika were endemic. We worked with the NIH on developing a heroin vaccine with NIDA. We worked with Dr. Kathy Newsel at the Center for Vaccine Development at University of Maryland on trying to develop universal flu vaccines and influenza human challenge models. We work with both biotech and big pharma to develop prophylaxis against dengue and chikungunya, mosquito-borne diseases, as well as HIV therapeutics. And then we work with non-governmental organizations like the Trudeau Institute and the Adirondacks on problems like Zika. But we also create actionable knowledge products. So that's Dr. Saravan and Tangamani on the right, who started a citizen tick testing program. And so from anywhere between March and November, we received thousands and thousands of ticks from all over the state. And he tests those ticks and determines whether or not there are pathogens, and that can inform local public health officials. And then we provide service learning opportunities to physicians who are in training, or even attending level physicians, to go to places like Machala, Ecuador, or Kisumu in Kenya, to broaden their horizons on what medicine is like to be practiced in other parts of the world. Next slide. But when the pandemic came, obviously we had to pivot. And so we basically, as Dr. Evans had mentioned, all non-COVID research essentially got put on pause, and we refocused all of our skill sets and expertise and our resources on the COVID mission. I'm just going to speak about the vaccine component of it. And so we had to really scale our operations to meet enrollment demand. You know, we all work with a sense of urgency, but clearly this was a much different initiative, and we were working with a sense of emergency, and it was very visible to us across the street watching the hospital fill with patients. Doing clinical research in the time of a pandemic, we needed to develop new safety protocols. We worked round the clock, not only with Pfizer and BioNTech, but the CROs and the regulatory agencies. This was a major recruiting effort, and the response was overwhelming. We're used to dealing with 50 to 100 volunteers, and within a week we had over 2,000 people that had called and wanted to be part of the trial. Endless data entries while we are sleeping. There are people in multiple continents who are reviewing the data that we put into the system and sending us queries, and so those have to be answered. That's a round-the-clock operation. And then, this was not just unique for us, but for anyone, being in the middle of a clinical trial and then having the FDA issue an emergency use authorization creates some complexities to the completion of that trial. Next slide. So what are we doing now? Well, we are unblinding placebo recipients who are eligible for vaccination according to the ACIP guidelines. And so by the 1st of March, all those 16 years of age and older who have been in the trial will receive at least a first dose, and that includes about 150 healthcare workers. We were one of the sites that enrolled 12 to 15-year-olds, so that cohort has now been filled. We enrolled over 70 at Upstate, 2,200 globally, so that data will be coming forth soon. And then we continue to follow volunteers for natural infections, and of course, we're very concerned about these variants. I put this image in here from the CDC a couple of days ago, and it's already outdated in the sense that we now already have over 900 cases of the UK variant, and with 34 states reporting. So these trials are more than 18 months in duration, and one of the reasons is to look for enduring safety and efficacy. Next slide. So we're also beginning new collaborations on other COVID vaccine candidates. So GM1 Life Sciences, which is a group I worked with when I was in the Army to make a MERS-CoV vaccine. We're now working with them again to make a novel COVID-19 vaccine. Again, additional work with Dr. Koloff at the University of Maryland. We're part of the NIH VTEU system, which of course is supporting Operation Warp Speed. And then we're starting to ramp up sort of the delayed or paused non-COVID work. So again, we're going to try and start this heroin vaccine trial this year, the pursuit of universal flu vaccine, looking at therapeutics for dengue, looking at therapeutics for Staph aureus bacteremia, and sort of getting back to our previous mission in addition to COVID. Next slide. So I really like this slide. So that is my boss on the left. That's Dr. Sri Narsapur. He's a nephrologist. He's the chair of medicine, so I work for him. And I appreciate that he supported my colleagues and I so that we could have the protected time to do this research. And he just received his second dose of the vaccine that we helped to bring to EUA. And on the right is his daughter, who's a fourth-year medical student that actually gave him that vaccination. So this drives home the big point for me for this past year, which is everyone trying to take care of one another. Thank you for your time.
Thank you, Dr. Thomas. Our last speaker is Dr. Diane Hernandez. She is a tenured associate professor of socio-medical sciences at Columbia University's Mailman School of Public Health. She is director of community outreach, engagement, and translation for 2 Columbia-based centers, and her research program focuses on the social and environmental determinants of health and examines the impacts of policy and place-based interventions on the health and well-being of socioeconomically disadvantaged populations. Dr. Hernandez.
Pleasure to be here. Thank you so much for the invitation and for the presentations from my fellow panelists. I am, so today I'll be kind of describing disparities by race, place, and neighborhood poverty, and also the kind of collateral consequences that are yet unfolding as this pandemic kind of has taken hold for quite some time. I'd like to note that I'm a sociologist by training and have contributed to socio-medical research at Columbia's Medical Center. And that's an important complement to the kind of work that happens in biomedical research, in part because it starts to at least kind of shed light on some of the challenges that specific communities are facing. So I, like I said, I'll focus my remarks not just on the disparate impact of the pandemic, but also to these kind of social determinants of health. One thing that we absolutely can take away from this pandemic is that COVID is a public health phenomenon, not only because it's a potentially infectious disease, but also because it really has exposed the devalued status of Black, Brown, and poor lives. I'm dispatching from my home in the South Bronx, and one of the things that was really obvious just about a year ago, at least to me and those of us that are in this space around the social determinants of health, is that in fact, as this kind of pandemic played out, we would probably see this kind of usual story around which communities would be kind of most impacted. And that was actually true. I wrote an op-ed in the Daily News back in early March of 2020, basically kind of noting that it wouldn't probably be just a question of older age that would determine who was most vulnerable to the pandemic, but in fact, that some of these other kind of issues that we have come to understand and appreciate within this kind of social, the social and behavioral sciences would unfold. And in fact, it was the kind of COVID color line, as I like to describe it, that has unfolded in the time, since. And so I'll be going over some kind of quick data points and then giving some background for how this actually all has come to unfold. So in terms of cases, we see that there's a disproportionate kind of representation among Latinx and Black or African American New Yorkers. This is New York City based data relative to their proportion of the population in the city. And this is just in terms of cases. So why is that? Part of it is that social distancing, has been considered a privilege. That in part is because of the essential workforce being comprised primarily of African American and Latino workers. And that many of them are in face-to-face occupations that haven't translated into the remote work world that many of us are privileged enough to benefit from. There's also the reliance on public transportation and also crowded homes, so doubled and tripled up housing, and what that ultimately means for the transmission of COVID. So this is part of the kind of explanation for the racial disparities in COVID. Then we think about hospitalizations and severe illness. As you see, this is kind of another metric around poverty level, so that very high poverty and high poverty communities were much more impacted by severe illness and hospitalizations. And that is rooted in some of the disparities that we've also understood pre-pandemic, right? The issue of whether or not these kind of higher poverty populations are insured. We know that we have some of the highest uninsured rates among the poorest populations in New York City. That we also have some of the highest rates of avoidable hospitalizations among adults and children in these poorer communities. And many lack a regular provider and encounter kind of overwhelmed medical facilities. So all of these things kind of also adding to the severity of hospitalizations. But another piece to this is also about the underlying comorbidities. So we know that hypertension, diabetes, and obesity kind of have led to more severe outcomes in terms of COVID. And when you think about that from a place-based perspective, or from the perspective of who's kind of more disadvantaged in this regard, you see that a place like the Bronx has higher obesity, higher diabetes, and higher hypertension. And in the kind of in the most impoverished areas, that's absolutely true in terms of New York City. And just by kind of for comparative purposes, you have, 2.5 to 8 times more prevalent, a higher prevalence rate in places like the Bronx compared to lower Manhattan areas that are marked by a much more privilege. Now, when it comes to death, you see that places in the Bronx, or places like the Bronx, as well as Queens, and specific areas in these boroughs have been most impacted by a higher death rate as a result of COVID. Really kind of, demonstrating the kind of place-based impact and that cumulative burden that communities of color, low-income communities are facing in this current pandemic. Now, we've turned the corner, and for the past 2 months, we have had the vaccine available. And the good story is that, we have over 1 million New Yorkers that have had at least one dose of the vaccine. But we also are seeing in kind of early kind of analysis that the racial distribution of vaccines, so these are adults that have received the vaccine as of, I believe it's this past week. So the overwhelming majority are white. followed by Asians and Latinos, and then African Americans. And if you recall the first slide that I presented around the racial distribution of COVID infections, you see that there's kind of a disproportionate representation of whites and Asians in this pool, and an underrepresentation of Latinos and of Hispanics and African Americans in terms of who's getting the vaccine. And this is really critical because, of course, we know that they're having the worst outcomes, but they're also kind of less engaged when it comes to the vaccine efforts. Some of that having to do with the misinformation that Dr. Fauci kind of pointed to earlier, but certainly an area for kind of further inquiry and intervention from a public health perspective. And so now I'd like to really think about what does this all mean for the long term? Some of the communities with the highest unemployment rates, the highest poverty rates, the highest rent burden and food insecurity are also the ones that have been kind of disproportionately impacted by COVID. But ultimately, this may just serve to deepen some of those existing inequalities. And so from a social determinants perspective, I'll be talking about jobs, about wage disruption, and other factors. So this is the national kind of picture with some data from both Brookings and the Center on Budget and Policy Priorities. And what we see is that in terms of race, you have kind of higher job losses and income losses among Hispanics, and also this kind of other racial category as well as whites in this during the pandemic. But you also kind of importantly see that folks that were already kind of in low wage sector jobs were much more likely to experience these job and income losses. And this is especially true kind of as broken down by industry. And so you have the highest kind of impacts in terms of employment in these populations. In New York City, you see that the kind of racial distribution of these job losses or wage disruptions are happening among Blacks and Latinos. There's been a lot of discussion about the impact on women, but we also see that men are clearly impacted. That those earning kind of less than $35,000 and having less than a BA or a bachelor's degree are also disproportionately impacted. And naturally, that would kind of introduce some challenges around food and housing insecurity, and that we see kind of unfolding in terms of food insecurity in the past 7 days. And this is as of October of last year, the kind of latest data available. But we also see that housing instability is also introduced, not just for renters, but also these numbers are comparable for homeowners as well. And when we think about what this ultimately looks like in terms of the racial distribution, the story is really kind of familiar, where Blacks and Latinos are much more kind of impacted by trouble paying for usual household expenses, and that this is actually compounded. So that for households with children, you have a higher kind of representation of those that did not have enough to eat, were not caught up on rent than other households. And that really leads us to think about the kind of intergenerational impacts of this, and academic losses are one example. So African American and Latino children are much more likely than their white counterparts to rely on remote learning. And what we see as a result is that they're also kind of disproportionately impacted by academic losses during this pandemic. Some of it having to do with the digital divide, which many of us kind of imagined was a foregone problem, but in fact remains with us. And many of these students don't have devices or reliable internet access. And lastly, I'd like to kind of point out really this kind of reckoning around discrimination and systemic racism that was brought to life not just from the data suggesting this disparate impact of COVID-19, but also the kind of racial reckoning that was introduced by Black Lives Matters and the whole issue around addressing, police violence and other forms of structural violence against communities of color. Surely there was the stigma around, in certain communities, especially Asian American communities, around COVID. But as we really come to kind of see this unfold, this has also been a story about the usual suspects of disparities that at this point, from a public health perspective, is kind of necessary for us to reimagine. That's partly about the conditions, the social conditions that determine these outcomes, and also kind of issues around care and treatment and just, like a survivability in these communities. And so with that, I'll just say thank you, and I guess we'll have some time for Q&A.
Thank you, Dr. Hernandez, and thanks to all of the speakers for a very enlightening session. We have a lot of questions, and I will just let the audience know, because this is focused on research, we're going to focus particularly on questions that relate to issues of research. There are a number of questions that really are very specifically related to clinical treatment, and we may be able to pass these on to somebody else and get you an answer at another time. The first question, which any of them could answer, but I think that I would specifically ask Dr. Thomas's view of this, is do you view the pandemic of this magnitude to be a 100-year event, or should we expect that with climate change and increasing mobile populations, et cetera, that large-scale pandemics could become more frequent? Having worked through the current crisis, what can the research community do to even better prepare for future events?
That's a big question. We have been talking about and writing about and having conferences and meetings for a very long time about the epidemic planets one day aligning and having an event like the one we're involved with now. And so, this is not a new concept of pandemic preparedness. But it does seem that we were caught a little bit flat-footed. My opinion about going forward from here is that, I think that, the federal government, the state governments, and academic institutions, and even the commercial space, the pharmaceutical space, I think that they have to make enduring and substantial and real investments in biopreparedness. And these investments cannot be tied to sort of annual appropriations. They have to be strategic, and they have to be long-term, and we have to invest in people. We have to get people interested in doing this kind of work. And by having those sorts of investments, we can create collaborations and networks that this is their job, this is their mission, to be prepared and to be able to identify and respond quickly. So I'm hoping that this is a 1 in 100-year event, because we learn a great deal and we make some changes.
Great. Thank you. Liise-anne, did you want to make a comment on that?
Yes, just very quickly. From the sort of pathogenesis point of view, we learned something from this that Dr. Fauci mentioned that we did not know, which was the transmissibility from people who were completely asymptomatic. This really, it was a new discovery for us, and I think really puts the spotlight on epidemiology, public health, and surveillance in terms of thinking about this type of a possibility in the future. By the time we had all of these sick people, the cart and the horse were fairly separated.
Thank you. Dr. Hernandez, maybe for you, there are a number of questions, but I think maybe if you could address 2 quick ones. What is the impact of school shutting down on the racial inequality related to this disease? And what kind of state policies could be enacted to lessen the health disparities based on social determinants?
In terms of the academic losses, it's kind of what I was describing before, that nationally at least, the majority of students, white students, for instance, are much more likely to actually do in-person learning, and that has a lot of implications, not just for the kind of, let's say, absorption of information and learning, but also for kind of the social emotional development of children. All of these, and the ability for parents, in fact, to be able to kind of keep up with their other obligations, such as work. And so obviously there is the kind of academic benefit of in-person learning, but also the kind of alternative to that is the fact that you have, like I mentioned before, African-American and Latinx households that are less connected to the internet, that do not have reliable devices. There's a disproportionate number of those students that are actually relying on phones, on cell phones, to be able to connect to school. We know that that's kind of inadequate. Any of us that have tried to just have an email, a significant email exchange on our phones know that that's actually very complicated. And so that for sure is an issue to kind of look forward to. And there's been kind of more recent discussion around, reopening schools, in part because a year of, or over a year of academic losses can have implications for completion of high school, for instance. And then that has implications for whether or not these students end up going on to college and having kind of fruitful careers in the future. And that, it starts really early, but has implications for the life course, essentially.
Could you comment also one quickly on how the homeless get involved, get placed into this whole issue?
Well, I think part of the kind of homelessness question is around homelessness prevention, right? So we have eviction moratoria at the city level, at the state level, and we also have that at the national level, which to me is an important kind of prevention strategy around homelessness. One of the things that has been happening in New York City specifically is that, they have been using non-traditional housing spaces, so hotels, for instance, and other opportunities for people to, maintain social distancing to some extent as alternatives to congregate settings for the homeless. It's not enough, and I think, there are so many kind of factors that, kind of contribute to a homeless population having greater, risk around this pandemic, but it has been one of the strategies that they've used is to kind of introduce different modes of housing folks that are homeless.
Thank you. Dr. Evans, you described a very exciting approach using stem cells to try to identify targets. And one of the questions is can you elaborate, can this be used more broadly? Where do you see the stem cell research going to try to identify potential therapeutics in other diseases?
Well, you're right in my lane on that question. I'll tell you, as a stem cell biologist, it's my favorite topic. It's the thing that I love about stem cells as a platform is that really you can study any disease. The great thing about stem cells is that we can generate any tissue types from them. And then, for example, with iPS cells that we can derive from patients that have specific genetic variations, we can model diseases that are specific to a patient even. So there's no limitation on how we can model diseases and use those models to find therapeutics or understand the underlying genetics behind the diseases. We're really in the early days, and that's why investment in this area is so important, because all these models, for example, the ones that I showed you, are pretty simple. So now many people are interested in starting to make these more complex models, starting to add in. So for example, one of the things that we are doing is in our cardiomyocyte models, adding in components of the immune system so that we can really understand how these different cell types interact with each other. So currently, of course, you may have heard of interesting and exciting concept of cellular therapies. So my neighbor across the street, Lawrence Studers, is trying to treat dopamine deprivation and neurodegenerative disorders using cell therapies. Macular degeneration in the eye can maybe be treated with cell therapies. But those are really, really challenging. Right now, we can actually pretty straightforwardly use these models to look for therapeutics like I described today.
I think we have maybe time for one or two more short questions. Dr. Pirofski, maybe you can take this. I think there's a lot of concern that we've heard about variants, viruses, and I guess one of the questions is, do you think that as these variants become more prevalent, will this affect things like the kind of treatment you're talking about with convalescent serum? And also, will this affect the whole approach? You know, are we looking towards having to have a treatment every year, or are we, you know, what's our concern?
Well, thank you for that question. It's a hard one. So I'll take the second part of it first. Yes, the direction in which we are heading is right now that we probably will need to revise our therapeutics and possibly even our vaccines on a regular basis to adjust to evolution of the virus, and much like we have done for influenza. Dr. Fauci mentioned the search for a universal vaccine, and I would say that that also could apply to universal therapies. It's very interesting that some of us seem to have forgotten basic virology. Viruses evolve. Our RNA viruses evolve a lot more efficiently. We have a huge viral load in the globe right now, and that's why we are in a race to get people vaccinated and to try that, bring that down. But as long as we have as many infected people as we do, the virus is going to continue to evolve. How that will affect convalescent plasma therapy is that recent plasma will probably be the plasma that we need to use. I will point out, however, that when people recover from an infection with the variant, they've made antibodies to that variant. So our ability to deploy convalescent plasma is really going to depend upon surveillance and the ability to detect variants and then to be able to identify people who have antibodies to them, all of which should be doable. I cannot underscore enough the importance of surveillance, supporting surveillance. You know, last year it was testing, this year it's surveillance. We are just not sequencing enough virus, and we really do not have a robust enough sense of how prevalent these viruses are. But bottom line is, yes, we will need to adjust. Yes, this is what viruses do. And convalescent plasma actually has a lot of potential to help us deal with variants.
Thank you very much. I think we are unfortunately out of time. There are a lot of questions, and what we will try to do is cull these questions and perhaps distribute some answers over the next day or so that everybody gets a chance to have had his or her question addressed. I would just like to thank, in closing, all of the panelists for really a revealing set of discussions. Also thank Dr. Fauci and thank our audience for participating. I would take away a number of things from this, and particularly echo what Dr. Fauci said about the speed at which science really has tackled an enormous problem, our greatest pandemic in 100 years. And I think here you saw the breadth of this. New York State, of course, was the epicenter, and in some ways still the epicenter of COVID in the United States. But what you've seen is all of the institutions throughout the state working collaboratively, getting together to really do research in many, many areas, both basic fundamental research leading to therapeutics, to helping to develop the vaccine, and psychosocial research, trying to address the issues of healthcare disparity that were only enhanced or made worse by an epidemic like COVID. But I think that we have to be very proud of the way the science community has come together collaboratively to tackle this. And I think it in a broad way, and I think it speaks well to New York State. I would also note the flexibility of the science. You heard many people basically say that they stopped what they were doing and pivoted. I kept hearing the word pivot in order to really address COVID. And that was certainly true in my own institution, where everybody who possibly could impact on COVID started to work on COVID. That's why you get 100,000 publications in such a short period of time. And the last I would say is we've learned some really very exciting possibilities in the future, both in terms of the use of stem cell research, which will have huge impacts, but in general on how science comes together to tackle the fundamental social and clinical problems. So I think we have a lot to be proud of. I think our speakers have conveyed it to you. I thank you all, and hopefully the next time we meet, it will be to tell you how we have put this problem completely behind us and can work on the wonderful problems that all of these investigators were working on before COVID. So thank you again.
I would just like to say one thing, if you don't mind, in closing. All of the presenters' slides will be on our website, which is www.amsny.org. You can find their contact information there as well. So if you have any questions that you would like to ask them directly, you'll be able to do that. And I would also like to thank Dr. Taubman, all of our presenters, Dr. Choi and Dr. Fauci, for taking the time to inform us all about this subject. And I just also would like to thank the staff at AMSNY and at Columbia who have helped with putting this on. So thank you all and have a good day.